right now in the company. So we are both producing some antibodies that can be used for research and diagnostics. And we have already 3 partnerships with 3 different clients to produce their specific antibody for such kind of application. Meanwhile, we are developing an antibodies, which is also industrial demonstrator, that is targeting particular disease, which is called neuroblastoma, which is a pediatric cancer that affect 25,000 children worldwide every year.
Welcome back to the Smart Biotech Scientist. I'm David Brolman, your host, and this is part 2 with microalgae pioneer Muriel Bardor. Now we'll continue our fascinating exploration of how microalgae are revolutionizing antibody production and achieving that impressive 70% cost reduction. Let's continue our conversation. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brolman, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Now I'd like to change gear a bit. I want to touch upon the quality side. And there are 2 things that come into my mind. First of all, let's start with the contamination. How does that work with microalgae, or how likely does your cell culture process contaminate, and why does it contaminate compared to a traditional mammalian cell culture?
So what do you put under the term contamination? Are you thinking about virus or mycoplasma, or what do you...
It could everything. It could be virus, it could be microplasma, it could be bacteria, it could be... let's start with these.
So as mentioned with a virus, so we work with an expert in virology, tested that there is no virus from human that can grow in the condition that we are using. So I will say we are virus free on our process. And of course, we run some analytics in order to prove that it was the case. We did exactly the same for mycoplasma. So we have some evidence that we do not have any mycoplasma in our culture. But again, I think there is nothing in the media that could be sufficient enough to grow mycoplasma. So it's the same in terms of bacteria. We are doing regularly some bioburden assay like the pharmaceutical company are doing, and they are always negative. But I think it's the same. We are not using any sugar, for example, in the culture media. So we have nothing that can be used as a primary metabolite for these bacteria to grow. And after that, if we are extending the notion of contamination, of course, we are working on the identification of potential host cell protein, which of course has to be followed during all the process. So this is ongoing work that we are leading right now in collaboration with a French company from Lyon, which is called Anacan, which is using mass spectrometry in order to identify and follow host cell protein. Because probably if you are familiar with the process with mammalian cells, they are using ELISA kits in order to follow the behavior and the way that the host cell protein are disappearing during the process. But of course, as we are so innovative, there is for the moment no ELISA kit which are commercially available for our bioprocess. So we decided to use mass spectrometry, which is a very powerful method in order to identify and follow HCP, and to start with mass spectrometry. And maybe in the future, we can develop our own ELISA kits, but that's going to be for future steps.
Since you mentioned HCP, so host cell protein, let's also talk about the second aspect of the quality. Are the in-process controls, so the analytical assays, very similar to the traditional process, or do you need to measure some other parameters?
No, I will say they are basically similar. I mean, we need to, of course, we start discussing with a French regulation agency, the ANSM. So we have some ongoing discussion with them at the moment. But the way we are processing right now, it's quite similar. I mean, we have planned, as I mentioned, to follow the HCP. We have planned to follow the putative residual DNA, which is using the same methodology as what is currently used for mammalian cells. So we are planning, of course, to keep the bioburden and the mycoplasma test in our process. So it's basically the same analytical part, except that we are not following the virus contamination because we know that for sure we are virus-free.
And how is it with respect to the regulatory aspects? I imagine you have already some discussions with agencies. How is this new technology perceived?
So actually, we have submitted already our project to the French agency, and we are supposed to have our first meeting with them in a few weeks from now.
Excellent.
But I'm quite optimistic with that since now a couple of years there is one product which are on the market. So it's not an antibodies, but it's an enzyme which is called the glucocerebrosidase, which is commercially available and commercialized now by Pfizer, but it has been already produced by a small biotech company in Israel, which is called Protalix. And this particular product is produced from carrot cells. So meaning that it's quite similar, I will say, bioprocess as compared to our macroalgae. We also read a lot regarding this particular product and the way they perform their analytics and they discuss with the regulation. So I think it will help us in the discussion as well.
And now, in the beginning, you've mentioned that you have been on this project for quite a while. So after more than a decade of research, you're now in the process of launching this commercially and scaling it up. So tell us a bit what you are doing right now and what will be the first applications or the first biotherapeutics you're going to test.
Yes, so just to make sure for everyone, so it's true that until 2021, so I was only working on this project as a professor from academia. And at the end of 2021, so we decided to create a company which is called Alga Biologics and that I'm leading right now, both as a CEO and as a scientific director. And the goal of the company was, of course, to do the valorization of the work that we have performed at the university, but was also to work in the industrialization of the bioprocess. So this is what we are doing right now. In 3 years' time from the beginning of the company, we managed to increase production capability up to 200 liters, which was already a big step. And we validate that we are able at this scale to produce some functional antibodies, which are presenting the exact same quality as what we had at the lab scales, which was not so obvious. And so
that's excellent. Yes, it's a challenge.
So we were really happy about this result. And right now in the company, so we are both producing some antibodies that can be used for research and diagnostics. And we have already 3 partnerships with 3 different clients to produce their specific antibody for such kind of application. Meanwhile, we are developing an antibodies, which is also industrial demonstrator that is targeting particular disease, which is called neuroblastoma, which is a pediatric cancer that affect 25,000 children worldwide every year. It's a cancer which is increasing, like all the cancer, unfortunately. And so far, there is 2 antibodies which have been developed and commercialized targeting this cancer. But these antibodies are produced in mammalian cells and they are very, very, very expensive. And they are not so, I would say, efficient as right now the kids which are treated with these antibodies have 50% of chance to survive. Since we can improve the efficacy of the antibodies, as we mentioned earlier, by increasing the ADCC response, this was a good first example of disease where we can bring something disruptive on the markets.
Yeah, excellent. As we look now ahead, so where do you see this going, and where do you see also the potential of the microalgae platform, let's say in the next 5 or even 10 years from now?
So I hope that we will manage to convince pharmaceutical company, which for the moment quite conservative, to change their way of thinking and to adopt this new methodology of production, which will not be so difficult as we are using some bioreactor to grow the cells. And so it's quite similar to what they are used to do with mammalian cells. And it's completely different than growing entire plants as I did in the future. So we hope to convince them through all the work that we have done already and all the good results. So we are already starting to discuss with some of them. And of course, they ask us to have some in vivo data on our first product, which is targeting the neuroblastoma. So this is the next step for 2025, is to get the pharmacokinetics and the pharmacodynamics on our antibodies to prove, and also to get some feedback from the regulators in order to prove them that this is not a dream, this can be reality, and that the people are ready to accept such innovative alternative. Because when you discuss, I would say, the people, they found this very, very great and interesting, and they are looking to new alternative and new solution.
Yeah, absolutely. Now, I would like to circle back to your story, because you've seen both the academia world as a professor. Now you're in the industry, leading a company, a startup, and developing an exciting project. What advice can you give to other C-level leaders and founders in biotech? Because, I mean, it's quite a challenge to first come up with a technology and then to commercialize that. What would you tell these people?
So maybe it will be an advice which will be more concentrated on my colleague from academia. They need to make sure that they have a lot of resource and a lot of competency that can be useful when you start a company. For example, how to look for money, how to manage very complex projects coming from academia. So you have a lot of resources in yourself and expertise and competency that you even don't realize before starting the company. But finally, you realize that it's very, very useful when you start the company. And of course, you are very resilient, because it's not so easy to work in academia, in research, at least especially in France. So you have this property inside yours that are very useful at the beginning of the adventure when you create your company.
Is there any way also to prepare for that, or you just have to do it and then you will see?
No, I mean, you need to feel it, and you need to be motivated by the fact that you are doing it. Of course, if someone is not motivated, it will not work. That's for sure. So you need to be, I will not say ambitious, that's not really the right term, but you need to get the pleasure to do it and to have the motivation to transform your innovation into a concrete solution and commercialized solution.
So now, from all we have covered today, Muriel, what is the most important takeaway you want our listeners to walk away with today?
So actually there is two. I mean, the first one is, when you want to try to develop your technology or your company, just try, so you will not have any regrets. And you will make whatever you can. And if it's not working, it's not working, but at least you have tried. This is the first takeaway message I would like to give. And the second one is that we need to pay attention to the ocean. This year is a particular year because it's at least in France, it's the year of the ocean that has been decided by our president. So there will be a lot of events to discuss about the role of the ocean and the importance to preserve the ocean, because as we mentioned, there is a lot of biodiversity within the ocean that will give us some key solution in order to cure new disease, for example, in the future, or to find new solution. So it's really, really important that the people are aware of that if we don't take care of the ocean and preserve the ocean, that will be a big disaster for the future generation. So please keep this in mind.
Excellent. Thank you so much for sharing this. So smart biotech scientists keep the ocean in mind and also just start and try. It's an excellent way to conclude our insight-filled discussion. Thank you so much, Muriel, for sharing these insights and also for innovating in this space where we need to make these amazing biotherapeutics accessible to more people. So thank you so much for making this happening. Before we go, where can people connect with you?
People can connect with me through email. They can find, I think, quite easily my email. There is also LinkedIn from the company and from my side. And of course, our websites. They can write us a message on the website and we will reply to them.
Excellent. So smart biotech scientists, there you have it. Reach out to Muriel, connect, learn more about this exciting new technology. And thank you once again, Muriel, for being on the show today.
Thank you very much, David, for the kind invitation and the nice discussion. Have a nice day.
There you have it, the blueprint for transforming antibody manufacturing through microalgae. Revolutionary advancements don't just come from incremental improvements. Sometimes we need to look in entirely new directions, even toward the ocean. I'm offering a free assessment to refine your CMC development strategy with precision. If this is of interest, book a free call with me through the link in our show notes. Until next time, keep innovating and remember, bioprocessing can be both scientifically rigorous and personally rewarding when done the smart way. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.